Hello, my name is Kevin Eberhardt, and today I'm going to be talking about an experiment and a study that my group did on varying levels of pH and its effect on the ability of the enzyme trypsin to hydrolyze the protein azocasein. But before I get into that, I wanted to discuss briefly what an enzyme is. So an enzyme is a protein inside of an organism that is used to speed up chemical reactions. It does this by lowering the activation energy of the chemical reaction. So the enzyme assists in the process by decreasing the amount of energy that is required from the organism in order for that process to occur. Now, this is very important for organisms because it allows us to speed up these processes and also to expend less energy in using them. So they are absolutely essential for life. In our case, we were studying the enzyme trypsin. Now, what trypsin does is that it speeds up the hydrolysis of proteins. This is a naturally occurring process. In us, it happens in our small intestine. And this is these large proteins being broken down into smaller segments so that our bloodstream can absorb them. Trypsin speeds up this process. So we decided to study the ability of trypsin to break down proteins at different levels of pH. Now, trypsin is, in our case, we studied whether or not it would break down azocasein, which is a milk-based protein that has had an azo dye added to it so that it's easy for us to read our results. And I will get to that in just a moment. But first, what was our question? Our question was, what is the optimal pH for trypsin to hydrolyze proteins? To catalyze the hydrolysis of proteins. And our hypothesis was that it would be at a pH of 8 because that is the pH of our small intestines. So higher than 8 and lower than 8, it would not be as effective at breaking down proteins. So in order to do this experiment, we used a substrate of azocasein. As I mentioned before, casein is a milk protein, and so we have added this azo dye with sulfonamide. Sulfonamide is added to the casein dye in order for us to better be able to read our results. So it forms this azocasein. And so that dye is absorbed at 440 nanometers in a spectrophotometer, so that when we can look at it afterwards, we can see how much of this protein has been broken down through the spectrophotometer by measuring absorbance. In order to conduct this experiment, we had 450 microliters of reaction buffers. So these are the buffers that are varying our pH. What we did was we did 3 repeats for each pH level. So we did levels 6 through 10, and we did 3 reactions using the reaction buffers for pH 6, 7, 8, 9, and 10. In addition to that, we also had a negative control. So in our normal reaction, we had the reaction buffer, we would add 50 microliters of trypsin, and then we would also add, so we add 50 microliters of trypsin to the azocasein substrate. In the negative control, we would add 50 microliters of water. So in that negative control, we're looking for no reaction to happen at all because trypsin is what breaks down the protein azocasein. So if we just add water, nothing should happen. And then what we did was we incubated this mixture for 20 minutes at 50 degrees Celsius. So we performed 3 assays at each pH, and then after the incubation period, we stopped the reaction by adding 10% trichloroacetic acid, 500 microliters of 10% trichloroacetic acid, or TCA. And what that does is it denatures the proteins and completely stops the reaction from occurring. So trypsin will stop breaking down. So we have this 20-minute period where we can say, all right, this is where the reaction's occurring, and that allows us to more easily compare the reactions at the varying levels of pH that we had. Then, as I kind of mentioned before, well, prior to that, we put them in we put the resulting mixture into a centrifuge, and centrifuge at max speed for 2 minutes. What this does is it takes out the big stuff. So it takes out the protein that has yet to be broken down, and it leaves in suspension this proteins that have been broken down from the azocasein. So the small proteins that have been broken down. Basically what trypsin does is it cuts these proteins between the lining and arginine points on the hydroxyl side. And so once we have these smaller amino acid chains, these will be suspended through the process of centrifuging. Everything else gets taken out. We can put it into a spectrophotometer, apply the spectrophotometer at 440 nanometers, and measure the absorbance. So the higher the absorbance, the more stuff that's in there. It means that our protein trypsin, or our enzyme protein trypsin, was more effective at breaking down the protein azocasein. So we found that the enzyme activity increased as pH increased to 8 and 9. So as you can see here, for example, batch 1, we have absorbance of 0.48 at 6, 0.638 at 7, 0.773 at 8, and then it goes back down to 0.730 at 9, and 0.447 at 10. So it increased to 8, and then it decreased after 8 for pH levels, the absorbance did, which is what we were expecting. However, we did have one outlier here. In batch 3, our pH of 9 had an absorbance of 0.09, which was not significantly different from our negative controls. So our negative controls are typically in the 0.08 area. In fact, we had one negative control that was 0.09. So for this, we can only assume that the reaction did not occur. Something went wrong in our experiment, and for this particular batch 3, pH 9, there was no reaction occur. So when we did our statistical analysis, we did not include batch 3 pH 9 because we considered it an outlier. It was clearly very different from the rest of our results. So we do have this clear, as we were expecting to see the absorbance go up to 8 and then down after 8. However, with statistical analysis, it was not, we did not have a statistically significant difference between pH 9 and pH 8. We did for all of the other levels, but between pH 9 and pH 8, we did not. So we cannot say that pH 8 was the most optimal or more optimal for the protein hydrolysis than pH 9 because there was not a significant difference there. So, moving on to the discussion. We originally hypothesized that a pH of 8 would be the optimal level for protein hydrolysis and thus would display the highest level of absorption. Our results partially support this hypothesis, as I mentioned before. We were not able to determine if between pH 8 and pH 9 was the optimal level. Now, in terms of whether this was the highest possible rate of enzyme activity, we believe that it was because we used a salinity of 0.15 molars of sodium chloride, and we also incubated at 50 degrees Celsius. And from our comparison with other groups, these appear to be the optimal temperature and the optimal salinity for this reaction to occur. So we believe that at 8 or 9, we do have the highest possible rate of enzyme activity. And so in comparison with other groups who did the same, they typically found that 8 was the highest, which we did find in a few of our results, but we could not say with certainty due to the lack of significant statistical difference. Give me 1 second here. Just wanted to make sure I covered all the points. So potential sources of variability here are that potential sources of variability are kind of user error, different things like that. Clearly we had some sort of error here with our pH batch of 9. So in future experiments, we would try and be a little more careful with that and make sure that we had 3 different results. And in a future experiment with more time and more resources, we would probably want to test an even higher level of repetitions than just 3. That's tough to do within lab time, but if we could do that, that would be great. So in conclusion, it appears as if pH 8 or 9 for us was the optimal level for trypsin to catalyze the hydrolysis of the protein. Thank you for listening, and I hope you have a great day.